Okay, so we are talking about all these different sequential stages, and in this picture you can see the complicated structure of the initiation of DNA replication. And you can see here that the addition of all these different enzymes, for example, at the very beginning, this is the enzyme DNA-B, because we are looking at the replication in E. coli. Now this DNA-B is nothing but, remember, helicase. So this is helicase enzyme. So helicase is now indefinitely in place. And what else we can see here is the complicated structure out there. For example, you can see this other units like this pink and blue colored units. This is the actual polymerase 3 unit. So these pink color units are the core enzyme of polymerase, and T, this means the tau subunits which hold both of the subunit of polymerase together. And DNA-G means nothing but the primase. Remember, because we require primer, and this is the primase enzyme. This is the primase one, and primase have added the primer sequence there. Which one is the primer sequence? Remember, the primer sequences are this sequence. This is the stretch of sequence that are added by this DNA primase. So this is the orange color sequence. This is the sequence which is the sequence of RNA sequence, not the DNA sequence, because they are added by RNA polymerase, which is a DNA-G or primase. Okay, and then it provides the 3' hydroxyl, and this is the stretch of the sequence which will be added by this polymerase 3, right? The new sequence strand. So in a place, everything is added, right? And also, if I zoom out a little bit of this image, you can probably see the single strand binding protein as well, as you can see it here. These are the single stranded binding proteins. You can also see them here. These are the single stranded binding proteins, or SSBs, as you can see, right? So they are everywhere, they are in the same place, and everything is going on all together, right? So that's how everything is now placed. Now the idea and the most important thing is how this replication will go on, because the polymerase need to add a sequence one after another. Now we have talked before about how DNA polymerase is added and how DNA polymerase hold on to both of these strands all together, and that's by the beta clamp and using the help of clamp loader protein. Remember, this is the direction of this chain to open. This is the direction of DNA helicase will act. This is the direction when helicase will act. So helicase will start opening the double stranded bond of the DNA at this direction, and the fork, the replication fork, this is the fork. Remember, this is the replication fork, and this replication fork move towards that direction also. Okay, and while doing that, it needs to synthesize both of the strands, both of the new strands. Now remember, the synthesis direction is also from this 5 prime towards this 3 prime. No problem about that. So as you can see here, in this case, these are the two different strands, because this is the parental strand 1, this is, say, the parental strand 2. So in both this case, the parental strand 1 and parental strand 2 is there. Now if they synthesize the new strand, that is in red in color here, this strand is itself from 5 prime towards 3 prime, and the direction of fork is also from 5 prime to 3 prime. So there isn't any problem of synthesizing this strand, and this strand can be synthesized continuously like that. So that's why this strand is called as, remember, continuous strands. Continuous, continuous strand, or simply it is also termed as the DNA leading strand, because the synthesis of this strand is continuous through the time, because the direction of this synthesis is same as the direction of the movement of the fork. But if we look at this strand, the parental strand 2, in this case, to synthesize this strand, we need to do the opposite orientation. Why? Because the parent strand is itself 5 prime to 3 prime, and the only way to add the complementary strand is 5 prime here, 3 prime here, right? So the directionality of synthesis, according to the rule of DNA polymerase polymerization, should be 5 prime to 3 prime, which is top to bottom here. But the fork is moving to the opposite direction. So that creates a problem for synthesizing this particular strand. And this strand is not being synthesized regularly like the continuous leading strand. So that's why this strand is called as the lagging strand, right? This is called the lagging strand. And remember, during all this process of elongation that we are going to see right now, all this process, all the problems that are going to originate is due to this lagging strand, because the directionality of the synthesis of lagging strand is opposite to the directionality of the fork and DNA helicase movement. So we need to have different strategies, different ways to figure out to synthesize that. Now remember another thing, the DNA polymerase can only add nucleotide sequences from 5' to 3', right? It can only add the nucleotide sequences from 5' to 3'. That means in this direction parallel to fork, right? And to synthesize this other strand, DNA polymerase have to move from opposite to the direction of the fork. But again, remember, the DNA polymerase is only having two different subunits for that, but those subunits are attached with each other by this tau subunit. So how could DNA polymerase manage to synthesize both of these strands simultaneously in the DNA? That is my question. So try to think about this question. DNA polymerase can synthesize the leading strand parallel to the direction of fork, right? DNA polymerase can synthesize only the lagging strand to the opposite or anti-parallel direction of the fork, right? So the problem could be solved if there are two different polymerase enzymes synthesizing two different strands. But the answer is no, that's not gonna happen. There is only one DNA polymerase 3 and both of the subunits are attached with each other. So there is no way that they are going to break down. They are going to add it together and they are going to stay as added subunits with each other. So how could this DNA polymerase 3 synthesize both of these strands as well as leading and lagging all together simultaneously during the DNA replication? And there is only one way to do that, and that's what we're going to talk about now. And that is the most complicated part about the DNA replication problem. So if you understand that part or that process, rest of the thing will be super easy for you, right? So let's Okay, so the answer to my previous question lies in this particular picture. It's a brilliant picture, and once you understand this, you will understand the process of DNA replication, right? And here we are not only talking about initiation, now we are talking about the DNA replication elongation. That means how the DNA replication will continue after initiation. Once the DNA primers is added, actually it's an RNA primer, so RNA primers we can say. So once the primer is added, 3' hydroxyl is in place. Now the rest of the job is to be uptaken by the DNA polymerase 3 enzyme. But there was a problem according to me, and I've talked about that problem in previous example, that the problem was to synthesize both the strands leading as well as lagging at the same time, because the fork or the replication fork movement occurs in this arrow, this direction. But according to the structure of the DNA, the way it can produce this complementary to the leading strand is obviously same like this helicase movement, which is the parallel to the replication fork movement. But the other strand, if we consider this other strand, we need to synthesize the DNA in this direction, that means in this direction, and in this case it is anti-parallel to the direction of the replication fork. So how come DNA polymerase as a single unit which is attached with themselves can synthesize both of these DNA strands like leading and lagging together simultaneously? Now the answer here is, the only way to do that is to bend the lagging strand's DNA in this way. So these are the two different DNA strands, right? So if this is the leading strand, this is the parent strand 1, this is the leading strand, we don't need to bother about that anymore. But for the other parent strand, which will help or which is acting like a template strand for the new lagging strand DNA, we need to think about it. And the only way to do that is to bend it in the opposite orientation. So instead of taking it straight, it's getting one circle like structure like this, one bent, one circle structure like that. Now if it does the circle structure like that, what it allows, now you can see here, this is the polymerase enzyme, right? This is the polymerase enzyme, DNA polymerase subunits, right? This is the tau subunit, and this polymerase is now attached with each other with this helicase and whatever. This is the clamp loader, and these are the beta clamps which are now hold in the tight right place to both of this parental DNA, right? So the only way to synthesize is the obviously the direction here at this direction, right? So why not bending the other strand in that orientation? So they bend that other DNA in this opposite orientation. So if it bends it like that, what happens actually? Both the DNA strands are now having the same orientation, same region or same direction of the DNA synthesis. How? Because remember the DNA synthesis begins. So let me zoom in a little bit here. Zoom out a little bit here. If you look at here, that the DNA synthesis begins from this particular area. It begins from this particular area, which is 5 prime. So the synthesis obviously will take place in this particular direction. But on the other hand, this other strand is not having that. So the only way to do it is to bend. So if it is bending the other strand of the DNA and start initiating adding nucleotides somewhere further, not at the very beginning, remember, somewhere very earlier region. So in that case, it can start adding nucleotide sequences one after another. So now imagine, just think of this model in your mind in an animated feature and figure. And now imagine this polymerase to move in the forward direction. What it will do, it will keep on adding nucleotide sequences in the same direction. Now you may think, how come it possible? How come it possible? In both these cases, if it adds its straight the same direction like fork in the lagging strand, it is going to be 3 to 5 prime instead of 5 prime to 3. But it's not. See here. If we look at this, this is 3 5 prime, remember? This is 3 prime, remember? Right? So the new sequences that are being added, obviously from 5 prime to 3 prime, that are being added here. It's just the trick of rotating or creating a circle like structure by the DNA template for the lagging strand. If it does that, it gets this scenario of adding nucleotides simultaneously in both the DNA strands together by the DNA polymerase 3, and that's how it achieves this particular task. That's how the whole process works like that. So what happens actually, once this process will be done, then again it shifts back and start adding. So how that process works, let's see that. Let's see the step-by-step process now. Okay, so now we can see how this elongation phase actually passes, right? For example, let me zoom into a little bit here, or let's look at here actually. So you can see here, this is the DNA strand, double-stranded DNA, which are the parental strand, as you can see, which is marked here. And the parental strand mean one is the red one, another one is the blue one. Both of them are parental strands. And you can see here, this is the helicase enzyme, right, which unwinds those strands from each other. And then the SSB proteins in green color, they bind with the single stranded structures there. And the RNA primers are added in different regions. For example, this strand, this green color section is RNA primase, primer, which is added. This is another primer section which is added. So these are the primers that we can see here, both this one as well as this one. These are the primers, right? And the DNA polymerase 3 enzyme is here, is this one. This is the DNA polymerase 3 enzyme that is in place, and they are attached with each other by the tau subunit which is placed there, right? So this is the overall structure. Now what we know about continuing both of the strand synthesis simultaneously, they need to take one strand and form a loop, right? Here we can see the formation of loop, and this is very important structure, because this is the only way for this DNA polymerase 3 enzyme to carry out the reaction very fast, right? So now if we zoom into it a little bit, let's say, it's having some kind of issues with autofocus. 1 minute. So let us focus it a little bit. Okay. So if you see here that here this is the formation of loop, and that is very very important to have a loop structure right there, right? So in this loop, try to understand these two images very closely, very very carefully. What you can see here is that you can see here that this is an RNA primer that is placed. This green color is the RNA primer which is placed here. Similarly, you can also see the RNA primer that is also placed there, right? Now, as the formation of this loop, this is the beta clamp loader that is placed there, a clamp loader protein. So imagine that using this particular strand, this blue strand as a parent, as a template, the extension of this red strand will occur. That's why there is an arrow over there. The extension of red strand will occur from 5 prime to 3 prime, which is in the forward direction, parallel to the direction of the fork movement. But on the other hand, this other strand, this red strand, is not having this parallel orientation with the fork, right? So in this case, they form this loop so that they can continue this replication again from this 5 prime to 3 prime, because if you look at here, this is again 5 prime to 3 prime direction. So how they will do that? They will do it because this polymerase will only migrate straight in this direction, right? And as it is migrating this direction, this polymerase will keep on adding nucleotide sequences, and then they start to form DNA sequence there. And they start to form this DNA sequence until and unless they soon get into the region of the primer. You can see the primer is added here and also there. So it starts from that primer, start adding nucleotide sequences, and when it reaches the other primer, previous primer, then the beta clamp dissociates, as you can see in the bottom picture. So you can see here the beta clamp, there is the beta clamp. The beta clamp now dissociates and the core enzyme is now placed, and they release this lagging strand. But remember, in the leading strand there is no release, so no release in the leading strand, because in the leading strand they keep on attached, this clamp loader keep on attached with this leading strand, right? Because they don't need to release it, because they will start on keep on synthesizing this DNA, right? Similarly, in this second case, what they do does actually, they take that primer. Remember, now as they synthesize this part, they get this DNA in hand, but now they take this as a primer, right? This was the direction here, this as a primer. So they take this primer. Remember, this is the arrow or direction of the primer. They take that primer here, and a clamp loader protein comes in and load itself there. So this is the clamp loader protein. I mean, this is the beta clamp, not clamp loader, actually beta clamp protein that loads itself first to the single stranded DNA here. And then the clamp loader protein helps this beta clamp to load and attach with this subunit of DNA polymerase 3. And after that addition, they'll again start synthesizing this DNA continuously like this until they meet this point of other previous primer. And then again they will release this, and again they find another primer and keep on doing this, because helicases keep on unwinding it in this direction, and remember the fork is moving at this direction, and also the synthesis is going on in this direction, 5 prime to 3 prime. And that's true for every different aspect of DNA replication. Okay, so now let's talk about how the actual process of DNA polymerization works there, right? So if we talk about it, if we start talking about this process of polymerization, So what we have here, let's say this is the growing chain, right? This is the growing deoxynucleotide chain or DNA chain, the backbone. Remember the phosphate linkage that is placed there between the different base pairs. And what we require for a polymerization to occur properly, we require one triphosphate. That's what we have here, one triphosphate, this one. And we also require one hydroxyl group, one free 3' hydroxyl group. That's what we also have here, right? So one triphosphate free, as well as one hydroxyl group 3' free, right? So as we have these two things, now the second stage of this reaction. So let's see the second stages of reaction. Let's slide it a little bit here. Here it is. So now remember, let me zoom out a little bit. So here now this phosphate group is present here, and also the hydroxyl group is there. The idea is that the hydroxyl group here acts as the nucleophile towards this phosphate, this first phosphate, that is alpha phosphate, that is placed here. So when hydroxyl is attacking this phosphate, this first phosphate, other two phosphate groups are released as PPI, and as a result, that new phosphate, I mean the new DNAs, I mean the new DNTPs or nucleotide triphosphate or whatever it is, the new DNTPs is added. They are added to the growing chain, and that's what we can see here. They are now added to the growing chain here, right? The same thing will happen. So if I zoom out a little bit here again, if you look at here in this picture again, so this was the DNTP. Hydroxyl attacks this phosphate, two phosphates are released and they are now attached with each other. So now they have another, previously there were three nucleotides, but now this is another one. So for this polymerization, they definitely need a free 3' hydroxyl as well as 3 5' triphosphate to occur properly, right? And also in this case what we see is breakdown or hydrolysis of the bond. So similarly, as they have formed the bond, they can also have the breakage of the bond by the hydrolysis process. So this is the set, and the water is required for hydrolysis. As a result, it will break down this phosphodiester bond here. As a result, it will get one nucleotide apart and three set of nucleotides are now in place. So this is the polymerization, this is the hydrolysis that can be found in different stages of DNA replication.